Hi everyone, I think we're ready to get started. So before we begin, I would like to acknowledge that the land on which we gather is the unceded territories of the Coast Salish people, including the territories of the Musqueam, Squamish and Tsleil-Waututh nations. Second of all, to welcome you here today to the Vancouver Aquarium and Oceanwise initiative. So thank you all very much for coming. Just to introduce myself, my name is Ruth Sharp and I'm the manager of community events here at Oceanwise. So our mission here at Oceanwise is to inspire both the local and the global community to become ocean-wise. So what we all try to do here is we strive to increase the understanding, wonder and appreciation of our oceans, empowering all of us to help protect our ocean health and our planet as a whole. So thank you all for coming along to listen to the lecture today. But before we get started with the main event, just a couple of small housekeeping things. Just to let you know that there's some events coming up, but the next lecture is in November. So we're going to be getting a talk from one of the plastic researchers here at Oceanwise. So if you're interested in hearing when registration for this event opens, you can go online and subscribe to get email updates, and in addition to hear about any other upcoming events. So there are a few in the pipeline, they've got to be confirmed, and then if you're on that list, you'll hear as soon as the registration for those are open. And second of all, just in case you didn't know, as you can see, we have a couple of cameras here. The majority of the events that I run here at Oceanwise are videoed and live streamed to the Vancouver Aquarium YouTube channel. So if forever you can't make a lecture, you can catch them online. And in addition, when question and answer time comes towards the end, I will have a microphone. So I'll come to you with a microphone for you to ask questions, and that's so that your question could be heard for the people on the live stream or watching the video later on. So to try to make things as smooth as possible, well, one person, when the one question is being answered, if you could raise your hand and I could come and find you so that you're all ready to ask your question when it's time to ask the next question. So yeah, that would be fantastic. And also, as you all know, all these events that I run here are free, and I make them free because I think it's important to make these educational lectures as accessible as possible to as many people. But however, they do cost money to run, so if you do have a few spare loonies in your pocket, it would be amazing if you could drop them in the donation box just outside, and anything that you give will allow us to continue running events such as these. So thank you very much. And now, for the main event, I'd like to introduce Dr. Elisa Germann. So she is right now a postdoctoral fellow at the University of British Columbia, and she's working with Dr. Chris Harley. She received her PhD from the University of Georgia's Odum School of Ecology, and the work that she does at the moment is looking at ecological theory of host-parasite interactions to better understand the fitness effects of parasites on their hosts in a rapidly changing world, which I think means how parasites can affect organisms as the climate is changing. So let's welcome Elisa.
All right. Oh, yeah. Good evening, everybody. I'm so excited to have everybody here to learn about sea star wasting disease. So my goal tonight is to give you an update on everything that we know so far about this wasting outbreak. And in service of that goal, I'd like to try something a little new that I haven't done before, so we'll see how it goes. But my intention is to give you to tell you a story, And for that to work, you guys need to follow along with my story. And so I want to make sure that everybody's following along. So what I'm going to ask is that if you find at a point that I've gotten somewhere that you're not following, that just doesn't make sense, or I missed something, I didn't explain it well, could you put your finger next to your nose? And I'm just going to watch, and I'm going to try and make sure I notice when that happens. And if I see enough fingers by noses, I'll stop and go back a little bit, and we can talk about, maybe re-explain what I was just talking about, because maybe I missed some important part. That's all right. Okay, so with that, I wanted also to start by checking in with you. How many people in this audience have seen a sea star in the wild? Awesome, I thought so. We're in Vancouver, they're all around us, it's fabulous. How many of you were here in 2013, 2014, and maybe saw some either wasting stars or noticed a change in the number of stars? Yeah, absolutely. So for you guys, at least at this beginning, I'm going to replay a bit of a part of what you experienced. And for people who maybe weren't here or didn't notice that it was happening, I'm going to give you an introduction into what happened. So we're going to start from a place where we don't know what's going on. So how this started, what we're looking at here, actually, we'll start with that, is a whole bunch of ochre stars. And they come in orange or purple or some sort of combination of orange and purple that's sort of these weird brown colors. And they like to aggregate. They're found both in the intertidal, so just on the rocks on the edge of the ocean. You can walk down and see them when the tide goes out. You can also find them subtidally. And what happened was around the fall of 2013, scientists were out looking and counting these stars, which is one of the things they like to do. And they started to notice really weird symptoms. So what we're looking at here are symptoms of wasting. This is the early stages. And it's just this sort of little lesion that you can see. And it's different from these little white dots. That's actually part of the sea star's normal anatomy. But these little lesions here were new and different. And if it was just these lesions, that might be okay. But eventually, as the lesions started to grow, we started to see them actually losing arms. So this is one arm's lost. This is several arms lost, sorry. And you can see, I mean, this is really sad and also quite disturbing. So these symptoms were looking just really unfortunate. And we didn't know why. We didn't know what was happening. Further, these are just the ochre stars, which were in the intertidal, but when we started looking broader, we also started to see them in the subtidal as well. So these are sunflower stars, and you can just see they're melting. This part here is sort of a bit of their organs coming out a leg that doesn't look good. And so these symptoms were really concerning us. And there were starting to see some really stark declines in these species. So you might have seen this amazing and terrifying picture. This was in early October of 2013. You can see this is an amazing rock just covered in sunflower stars. So right here, they're just all over the place. And this was at the early edge of 2013. By the late edge of that same, this is in October, in that same rock, there's nothing there. You might see a little bit of this white that could be some dissolved former star. So we're just seeing incredible loss, and especially for the sunflower star, really fast. And so obviously this was incredibly concerning. And I want to take a moment here and highlight that as we started to watch, as this started to happen, we were finding these occurrences in a really wide range of places. And amazingly, we had a huge network of citizen scientists and aquarists and just general anybody going out and reporting where they went to look for sea stars, if they found sea stars, and if they were sick. So I want to sort of show you this video here. And on the left, you're going to see in red, those are points where somebody went to the beach and found a sick star. On the right, those are points where somebody went to the beach and did not find a sick star. So this gives you an idea of how hard we were looking and then how many sick stars we found. Okay, so, and you can see in the upper left, time is passing. You can see that it started up in Washington and Oregon. We started to find some down in California. To some extent, that might be because we found it first in Oregon. We don't know exact timing there. And now you can see it's spreading. We're having wider and wider spread. It's going further north. And there's a noted lack of either blue or red dots kind of in the central BC coast, which is something we'll talk about. That's mostly because we didn't have anybody looking. Yeah, so it's starting to be pretty much the whole coast. And there's not a lot of spots where people were looking and we're filling in sort of up north. There's not a lot of spots where we're looking and not finding the disease. And a thing, yeah, okay. That is the whole video. And this was possible because tons of people helped. So that's amazing, and we know a lot more about what happened and where it happened because people were out looking and sharing that they were looking, which was amazing. As well as having the citizen scientists out there, the people who generally go out and count sea stars, so all the scientists that have long-term areas that we go back to year after year and count sea stars, we're also doing quantifications. And so this is the part that I'm going to show you next. And I just want to kind of give you a visual of what it looks like. This is sitting on a rock. We're sort of looking at the rock here. And you have this transect line, which is the white line that goes up. That's measuring out a set area. We set out bolts in the rocks so that we can find the exact same area again, over and over again. And we go out, and you can see we crawl around and look in every nook and cranny that we can and try and find every sea star that's there. And often we measure them as well. That's another thing that we do. And so that's generally the surveys. And these surveys have been going on, and you can see on the left here is, again, the coast, and all those black dots are areas that have long-term data for them. So it's actually a pretty amazing data set. Okay, so this is what the data that came from that is going to look like. And I'm going to start sort of by telling you that each of these lines is a single location. So we have Landing Cove, Sea Lion Rookery. Those are each one location that we've been going back to over and over and over again. Within each of those lines, the colors are relative just to that location. So if it's blue, it's at about the mean population before 2013. And if it's red, then it's lower. So if you look to the left, you can see that basically there were fluctuations. Sometimes it was blue, sometimes it was a little red in the populations, up until around 2013, 2014, when for all of these sites, it goes quite red. And one of the things I want to note is that these are on a log scale, so even if you're sort of orangish red, you're already at a pretty much like very close to 100% loss of the total number that were there. So these are really big declines. They're really quite disturbing. And so now that we've looked at a little piece, and this is of the Southern California area, I'm going to show you the whole coast at once. Okay. Oh, and it's a little blurry, but you can still see the colors pretty well. And what you can see again is that we've got a little bit of blue and reds going on throughout the time periods that we were looking. You can see that obviously we've been looking for longer in California than we've been looking up further north. There's a pretty distinct change that happens right after this outbreak. So we really were seeing a really broad scale decline. And this data, I didn't specifically say, is for the Ochre star. So this is the intertidal star that we were looking at. Okay, so obviously the burning question is what is causing this disease? And as I get into this section, I'm going to start and step back away from sea stars for just a second and talk about marine disease in general. And the reason I'm going to do that, I'm going to start by talking about a system that we study quite a bit. And this is coral. It's an Alcorn coral. And this one is actually growing in Florida. And we know that corals have a variety of different diseases that are associated with them. And there's a group that was looking at Alcorn coral from around 1995 until now. They still are looking. And they found there's a specific disease called white pox disease. And it's a disease that's fairly well characterized for a coral disease. These can be hard to do. And this is what it looks like. They're white parts of the coral. And it is caused by Serratia marcescens. And specifically for white pox disease, the definition of the disease is that you have the disease, you have these symptoms, and Serratia marcescens. Those two things together make the disease. But one of the things that they found when they looked at this disease over a long period of time was that the strain of the pathogen changed. So in like in the earlier stages of the disease, they had one strain, and towards the end, they had a second strain. But the actual symptoms, what we were looking at, stayed the same throughout. And so while it's still one pathogen, one thing causing disease, it is changing over time. And this caused them to create something called the moving target hypothesis. So this idea that we can actually have one symptom have multiple different things causing it. And they argue that this can happen, particularly in marine invertebrates and coral in their case, because the coral only has so many ways to look sick. There's only so many ways that we can look at it and tell that it is sick. And so you can have one symptom and many things causing it. And that sort of seems a little bit out of there until you think about some of these human diseases that we have. For example, pneumonia. Pneumonia is where you have fluid in your air sacs and causes coughing and phlegm, and there's a variety of symptoms that we know are associated with pneumonia. However, we also know that bacteria, fungi, and viruses can cause pneumonia for us. And so there can actually be a different relationship between the disease agent or the pathogen and the symptoms that happen. And so I am going to go back to sea stars. And I imagine at this point, you probably don't think I'm going to tell you a really straightforward, it's one thing for sure, story with that background and introduction. And unfortunately, that is the state of the knowledge right now. You may have heard a few years back that there was a virus associated with sea star wasting disease. And up here is the picture of the virus. It's a denzovirus, and it's called wasting-associated denzovirus, and it was associated with wasting in the sunflower star. And that means when they found sunflower stars that are wasting, they found this virus. And they did, so that was determined. However, this disease wasn't just in the sunflower stars. It is in a wide variety of different sea stars. So 20 different sea stars. This is just an array of sea stars, not all 20. And so we didn't know if it was this same virus in all of those different stars. And newer research that's just been done was looking for this same virus, see, do we find the same virus in the Ochre stars in the intertidal? And this was work done by Melissa Pespini and Melanie Lloyd. And they did this quite recently. It has been accepted for publication, but it has not actually been published yet. And I actually just talked to Melissa this weekend and got permission to tell you about this research. So this is very new. And what she did, I'm going to walk you through their experiment and then tell you what they found. So what she did is she collected these Ochre stars from the field and brought them back into the lab, put them in individual containers, and then they actually, a proportion of them became sick on their own. So they were already sick. They came into the lab sick but not showing symptoms. And so at the very beginning, nobody's showing symptoms, but we have a top line and a bottom line. And just to give away the storyline, we have the top line is going to stay healthy the entire time, and the bottom line is going to start getting sick. So this is just, she had a certain proportion that got sick throughout the experiment. And what they did during this time was take biopsies of the tissue and look at the microbiota that were on the tissue. So the microbiota are the small microorganisms that create a community on surfaces. We have them on our skins. And some of the microbes can be positive, some of them can be beneficial, and some of them can be negative or pathogenic. And so she wanted to look and see what happened with those microbes over time. And she also tested for the virus through this experiment. And then on top of that, she actually looked at what the host's genes were doing. So how is the host responding in both the healthy and the sick ones? And so as time progressed, so we're on day 3, you can see the top still looks healthy, the bottom is getting sicker. You can still start to see those lesions coming out. Now they're just, bottom is sort of just a pile of legs and they're dissolving. I'm so sorry, it's so gross. It's really sad. Anyways, so this is what happened in her experiments. And the first thing I'm going to tell you is that they didn't find any of the virus in any of these. So there was no wasting-associated virus in this experiment. So that doesn't mean there's no virus at all. It could be another virus, but it does mean that this particular virus that was associated with the sunflower star is not what's causing these particular ochre stars to die. But they did see some really interesting things in the microbes. So one of the things is that she found that the healthy stars, the microbial community stayed the same throughout. They kind of continued on and had a very similar community. In contrast, in the sick stars, they saw a loss of the beneficial microbes. So the microbes that are associated with benefit in a variety of different ways and are actually used as probiotics in some cases that were found on the skin, that number decreased. And she saw an increase in a variety of pathogens. So not one pathogen, but different pathogens depending on this individual star. Yes, and there were, so they say the same throughout, but they generally had more to begin with of the beneficial microbes. So the healthy ones had more beneficial microbes. And the sick ones had actually a decrease in their immune functions and the genes that are associated with cell integrity, so the ways they would hold their cells together. In contrast, the healthy ones had an increase in those genes that are associated with cell integrity and with their immune response. So those healthy stars had good microbes helping them. They were fighting with their immune system, and they were trying to hold their cells together. So it looks like they're really fighting off something, and in this case, succeeding. And really interestingly, she found that there were some genes that are associated with neurodegenerative diseases that were actually in these sick and dying stars. So this is all really new information, but it generally suggests, and this is sort of the outcome of this, that whether or not you enter this experiment and are healthy at the end or are sick at the end really depends on your microbial community. And so this is something that's really fascinating, and we're going to be looking more this direction. And hopefully we'll begin to get a better idea of what this disease is. Okay. Yes, and as I said before, up to 20 species were infected, so we've got a lot to study and think about when we look at this. For the next part, I'm going to talk a little bit about the actual effects, so what we saw happen because of wasting, and I'm going to talk about 2 different species, because one of the things we found is that each of the different sea stars actually has different responses to this disease. I'm going to talk about these 2 sea stars. So on the left, we have the mottled sea star, and on the right, we have the ochre sea star. And these 2 are actually found just around here, and thus the work that I'm going to be talking about, I guess just to remind you that this is research that was done looking in the intertidal. And the work I'm going to be talking to you about was done just right around here, just right around us. So these are all sites around Vancouver Island, and if you haven't already recognized where we are, that star is where we're standing right now. So this is all work that happened very close by. And what we did is we went out and spent years looking at the number of those 2 stars, the mottled star and the ochre star, that we found in the intertidal. And before wasting, so this is our historical data set, if you were to go out to a beach around Vancouver, what you would find predominantly is the ochre star, and you might already know that. So this is what we're looking at on the y-axis is the mean percent of species in the community. So what this is telling us is before wasting, 90% of the stars that you would find around Vancouver would have been this ochre star. And you'd find about 10% of the mottled sea star. And to some extent, we thought that this was because these two species consume the same thing. Both of them like to eat mussels and barnacles. And so the thought was that the ochre star was just a better competitor. They're better at eating the barnacles and mussels and keeping the mottled star out of the intertidal. So after wasting happened, we were interested in seeing if that had stayed the same. So we went back out to these sites. Oh, what is happening? Okay, great, going the right direction. And after wasting, we found a really marked change. So now we're seeing that around 60% of the stars at a site are this mottled sea star or Evasterius. I'm sorry, I realize that I have Evasterius and Peisaster written up on the top. Those are their scientific names. Evasterius is the mottled star, and Peisaster is the ochre star. But what we would find is that there were around 60% of the Evasterius at these sites, whereas previously there were only 10% of the population. And what we found through experiments in the lab is actually that while both of these stars are infected by wasting disease, so they both get wasting disease, they are affected differently. So the mottled sea star takes longer to get infected. Once infected, if it's going to die, it dies quickly, but not all of them die. A lower percentage of them die. Whereas the ochre star takes a little, is infected longer, stays infected longer, and very large proportion of them die that are going to be infected, at least in the experiments that we ran. And we think that this difference in disease response is actually driving the difference in community response afterwards. For these mottled stars, even though they're getting sick, having a decrease in their competitor is allowing them to increase their numbers. And that's why we're seeing so many more of them now. Yes. And so after wasting, we've seen this increase in the mottled star and a decrease in the ochre star. Okay, so the next sort of vignette we're going to talk about is what happens to the prey of these sea stars when so many of their predators die. And for this story, we're going to go back into the subtitle and talk about the sunflower star. And I just want to remind you that for subtitle work, it's pretty amazing. To get all this data, we have to dive. And so all of the next section, it's coming from diving teams and divers going out and counting how many stars they see underwater. And you can see there's a small juvenile, and they'll often measure them there as well. So what we're going to look at is the sunflower star. And the sunflower star consumes these sea urchins. And the sea urchins actually consume this kelp species. So we're actually going to look at 3 levels. We're going to look at what happens when you take out, when the sunflower stars decrease, because remember we saw those horrifying graphics at the beginning of, they had a really large decrease in numbers. And then what happens to the urchins, which is what they eat, and thus what happens to what the urchins eat as well. So this is a trophic cascade, if you've learned about that before. So first, this is the data showing the number of sea stars before and after wasting. And you can see that this is just a count to show what that picture was showing as well, that there were less sea stars after wasting. And then what they found next was that the things that they eat, which you might expect, increased in number. So there were many more urchins after wasting. And then what the urchins ate, which is the kelp species, decreased after that. So we actually see less of cover of kelp species. And this is something that we've been seeing across the coast. So this study was actually done again around here, but there have been studies done up in central BC coast and in California that have shown really similar results. So sort of to summarize in a cartoon, before wasting, we had a community that had the sea stars, it had some of the urchins, and it had lots of kelp. And after those sea stars were removed from the system, we ended up with more urchins and fewer kelp. Okay, so now we're going to start talking about a little bit more uplifting side of this story. And I'm going to tell you a little bit about evidence for resilience that we're seeing in this system. And in particular, I'm going to be looking at the number of juveniles that we find. So how are they recruiting? Are there more baby starfish around? And for this section of the talk, we're going to move up to the central BC coast. So where that circle is, it's just north of Vancouver Island, is an island called Calvert Island, and there's a research station called the Hackeye Institute, well, that is on Calvert Island. Hackeye. And this work was done out there. And it's quite beautiful, like our coastline here can be. And so before wasting, we were quantifying what proportion of the population was juvenile. So how many small stars do we see? And I'm sorry these pictures are sort of behind me, but they're very cute little stars. These pictures are maybe around pinky-sized, and they're just amazing. And they can be hard to see, but you saw we were crawling around looking as closely as we could. And before wasting happened, if we were to go crawl around, we'd find maybe around 5% of the population had juveniles in it. And it was actually quite rare to see juvenile stars, and they're sort of sporadic, and we just didn't really understand why, but they were really quite rare. What happened after wasting was really remarkable and surprising to everyone. There were tons of juveniles. And this is, again, proportion juvenile, so like how much of the population was juvenile. This is about half of the population that's juvenile. And this is amazing. And it's something that I'm showing here just for up at Calvert. But again, for those surveys that we showed at the beginning for mortality, they actually saw similar patterns in those surveys all the way up and down the coast. There are just a lot more juveniles than there have been before. And there are a lot of things that could be causing this to happen. And I'm going to say that we don't actually know exactly why there are so many. It could be that in the early stages of wasting, the adults recognize that something's going wrong and just reproduce a lot. And so they make lots of babies. That's possible. It could be that because so many of the adult sea stars died, maybe the adults and the juveniles were competing. And so maybe now that the adults are gone, many, many more juveniles are surviving once they've landed. We don't know, actually, the answer to that question, but we do know that there's a lot more of them. Yeah? Yeah, so they probably would get to around sort of loony size, maybe in a season. Yeah, maybe in about a season. So we would expect them to grow up. And these two, I have two different bars here, and I've binned them both as after wasting, but these are 2 different years. So we see sort of a consistent number of increased juveniles as well. Yeah? Oh, thank you so much for asking. These are the ochre stars. Yeah. I had one more over here. Yeah. Yeah, so that's the interesting thing. For Calvert Island only, the actual numbers appear to have gone up, but we don't have as much before wasting data as other locations, so I'm not sure, but the absolute number of stars was larger. Yeah, that's a great question. Other locations, that's not necessarily the case. Okay, so what we found was that after wasting outbreak, there was really high juvenile recruitment or survival. The other part of this particular stage is that we also have people who've been looking out for recovery or locations that look like they have started to recover. And it's first I'm going to give you a little summary, sorry. So the things we've talked about so far are that likely your microbial community, this is upper left corner, likely your microbial community is going to determine how you do and how you fare. It's going to help potentially keep you from getting sick, so the beneficial microbes are associated with sea stars that aren't getting sick. The different species of sea stars, and this is going lower left, have different responses to disease, and that's actually going to change sort of the community, the relationships between the sea star communities. Up on the upper right, we found that the change in this top predator can actually change the number of their prey items, which then can feed down into what they consume as well. And then we found this high increase in juvenile numbers across the whole area, which is hopeful. And again, I was just about to tell you that we do have some information about how the sea stars are recovering, so how the populations are doing at the whole scale. So this is starting on the left in 2015, and again, these are the data that go from California up to Alaska. And if there's a little red X, that's a site that's not recovered. If there's a black dot, that is a site that's in recovery. It's not recovered, but it's showing signs of this high recruitment and sort of signs of growth and that the populations might be recovering. And so 2015 is pretty much right after the disease outbreak for much of especially California, Oregon, and Washington. And so most of those sites are still not in recovery. By 2016, and again, this is for the Ochre star, by 2016, we're actually starting to see an increase in recovery, so areas that are starting to recover. And then finally, by 2017, that's actually increased as well. And if you look, we're seeing more recovery in the middle, and that is an area where we've seen that really high recruitment as well, and a little less so in the southern regions. Okay, so I want to thank you all for listening, and I want to tell you that the work that I was talking about today, the parts that I did, were either part of the Hackeye Institute or part of the Biodiversity Center at the University of British Columbia. And I also talked about other people's work. And then I wanted to also give you an idea of what you can do. So I talked about citizen science through this talk. And if you're interested in getting involved in documenting these events, there's sort of 3 different ways, and there's probably more, but these are the 3 I know to do so. So the seastarwasting.org is a site that you can go, and if you see a wasting event, you can take a picture, and you can send in an observation. There's also one out that's, I think, just beginning to get underway, which is on Strawberry Isle. So that's actually just is at Strawberry Isle, it's out on Vancouver Island. And so they're starting a program. And then there's also iNaturalist. And I don't know if you've used that before, but it's an app you can put on your phone. You can take a picture and report things that you see in the wild. And it doesn't actually have to just be sea stars, it can be anything. And it can actually help you ID those things as well. Yeah, so these are different ways to get involved. And on this point, while we are seeing areas that have sites of hope and recovery, I can also tell you that the disease is not gone. So this last summer, we did, in fact, see wasting sea stars at a much lower level than we've seen before, but they are still around. So it is still important that we know where the wasting is happening. So this kind of information could be really vital. So yeah, and thank you for listening, and I'd love to take lots of questions.
I'm afraid the microphone's not working, so if you can repeat any questions
Okay.
until I get it working.
Yeah.
Hi. It looked from the slides that one of the effects of disease is that there's a lot less kelp. My understanding is that the kelp is a cover for juvenile fish of all kinds. So is the implication then that this sea star wasting disease has actually affected fisheries to a larger extent, not just commercial and fisheries, but all of fish at the sea and the aquarium?
Yeah. So that's a great question. I'm going to repeat the question. He was asking that his understanding is that kelp forests are a great nursery ground, so a great place for young juvenile fish. And so potentially the implications of having that kelp decrease could be quite bad for the fishery, for fisheries in general, and it's a decrease in the nursery. Yeah, so that's spot on. That is something that we're really worried about, and people are absolutely looking into and trying to understand. There's, so that, I guess yes is unfortunately the answer.
So all the sampling sites throughout North America, do we know, is it a global phenomenon, or does it continue down the coast, or do we know?
People are looking, it does not appear to be a global phenomenon. So my understanding is this outbreak is mostly just, as far as we know, been found predominantly on the West Coast. There's some suggestion that on the East Coast of the US, there were also some sea stars that died, and it could have been associated to wasting, but we're not sure. Outside of the US and Canada, my understanding is that we have not found any wasting. They are looking. So I've talked to a researcher in New Zealand who said that there is there's definitely people out looking. She said one person might have seen something, but that's pretty much it. So probably it's isolated to here.
Are juvenile ochre sea stars less susceptible to sea star wasting disease than the adults?
Yeah, so the question is, are juvenile ochre sea stars less susceptible to wasting disease than the adults? And the answer is yes. So in fact, it's a really striking pattern in all the different sea stars. The adults seem to be most susceptible. And in fact, sort of with the sunflower star, all the adults died, but some of the juveniles actually survived. And the same is true for the ochre star.
Do we know why that is?
We do not know why that is. Yeah. Mm-hmm.
Were any of the areas where they've been introduced and thriving, did they seem related to the drop in sunflower stars?
So the question is that sea otters also eat urchins and have an effect on kelp, thus through the urchins. And so has that sort of interacted with the sea star loss? And the answer is also yes. That there's a really great study by Jen Burt that was done up around Hackeye Institute as well. And they found essentially that the sea stars and the sea otters eat different sized urchins. And so, yeah, so the otters tend to eat the larger ones, and the sea stars tend to eat the medium-sized ones, or the smaller ones. And so there's a sort of a nuanced response. And they have an amazing data set where they actually have areas where the sea otters have come back, and areas where and then all the Pycnopodia died later. So there's a, yes.
So did the sea otters, in areas where there was a decrease in the sea stars, that they were starting to broaden their selectivity
Of size?
Yeah.
So the question is, did the sea otters broaden their selectivity of size for urchin prey? Yeah. Mm-hmm. That's a great question. I don't know of any evidence that that's happened, but that seems like, I'm not sure.
We have a question here.
So I want to ask that how it is actually affecting the ecosystem of BC.
Yeah, that's a great question. The biggest way for the subtitle is through the effects on urchins and the kelp forests. And then in the intertitle, we actually have less evidence about how the loss of the ochre stars is affecting the intertitle at the moment. However, we know they're a dominant predator, and they consume mussels. And so it's possible that there would be an increase in mussel cover as a result of the sea stars' decline. And that's not something that we have evidence for in BC yet. Yeah.
I just wonder about the virus itself. Did it have did it go up and down, and how much? Just don't understand. It wasn't
Yeah.
here before, it came.
So yeah, I'm going to repeat the question. Essentially, the question is essentially why is there an episodic time that we saw a lot of disease, and is it going to leave? And I wish I could answer that question. I think we're still in a state where we don't completely know what happened with the disease. So it could be that stressors, a broad scale, stressors on the C-star communities in general make them more susceptible to diseases that were already around. And the work by Melissa Pespini suggests that potentially sort of a change in their microbial communities just allowed other viruses and bacteria that were present previously to cause disease when they hadn't in the past. And it does seem to be associated with stressors, but we're not sure yet.
So would it maybe be increase in temperature of water?
Yeah, so the question is it related to temperature changes? And there is mixed evidence on that front. So we have several studies that have shown in the lab that the disease progresses faster and when it is warmer. So yeah, that has been shown. They've also shown that when you take an infected animal and put it in colder temperatures, the disease progresses slower. Both of those are lab studies. In the field, in Washington, they have really clear evidence showing that there's a correlation between increased temperature and disease prevalence in the summer that it outbroke. But in Oregon, they have the opposite pattern. So we're not sure yet, and it's possible that those sorts of, the differences in those responses and those correlations could come down to timing of when a stress event happened and a response afterwards. But that's absolutely, temperature is one of the main ones that we're really looking at and seeing if we can find relationships in all of the locations. That's one of the things that's really interesting and we struggle with this system is that it happened in such a wide space. So figuring out a consistent pattern between all of them is hard. Yeah. Oh.
Oh, no, this one here.
Oh,
Sorry.
Hi. Has there been any research done on whether certain pH or salinity levels increase the transmissibility of the disease or infection rates?
That is a great question. I'm just going to take a moment and think. So those, again, are really good hypotheses for stressors that could induce disease. I do not believe that we have any evidence that it changes transmission rate at this time, but that would be a great thing for us to look for.
Is it not unusual for a disease condition to cross so many species? And just in my experience looking around, there were two species that I always saw looking really healthy, and that was the bat star and the leather star. So did it touch those species? And is this interspecies spread really odd?
Yes, having so many different species all respond at once is fairly unusual. Yeah, and alarming, obviously. And you're right. So the leather star is one that is not. There is some evidence that it might be susceptible, but very much less than all the rest of the stars. And that is one of the patterns that we've started to see, particularly around Calvert, is the leather stars seem to be doing fairly well right now, and potentially some of their populations increased after wasting happened. So they may have also sort of like the Evisterius has been slightly affected, but mostly they were just benefiting from the increased amount of, or decrease in competition. And they did seem to be resistant. And there are several other species that are resistant and have different responses to the outbreak.
So are we studying them in
Yes, yes, that is absolutely one of the questions. One of the interesting things about the wasting, so the wasting-associated virus, when they go across species, they do find the virus, which, like I said, is associated with some cases of wasting, and some cases it's not. They do find it in non-wasting species, but they found, I believe, less in in the leather sea star. Yeah.
Has anybody looked at those early lesions? And in particular, if you were to biopsy, say, the interface between an early lesion and normal tissue, what is going on cellularly?
Yeah, so that is actually even newer research by Melissa Pespini. So the research I was talking to you about today is mostly from a paper that's in press, but there is another paper that she is working on that's looking at the RNA, so the gene expression at those interfaces in between healthy and diseased tissues, which means I don't know the answer yet, but someone does. And if you pay, hopefully it'll be published soon, and there'll be news about it, and we can hear.
I'll ask you to repeat. Is there any thought that microplastics might be affecting or acting as a stressor?
That's a great question. So the question is, are microplastics one of the stressors? I don't know if anybody's looking at that specifically, but it is a good question. So one of the hard things about a disease that seems to be associated with stressors is there are a lot of candidate stressors. And so we're working on sort of narrowing down which ones are related. But yeah, that's a great question.
Hey, I'm just wondering, has the wasting-associated virus actually been described before? And also, is there any record of like previous outbreaks?
Great question. There are wasting events so there are records of wasting events. We've not, I don't believe, okay, so there are records of wasting events, and when they went to historical samples and looked for wasting, so they got samples from aquariums and from museums, actually mostly museums, they did find wasting-associated densovirus in those museum samples, but not all of them. But yeah, so it's possible that previous outbreaks could have been from that as well. Okay, so the question was, where were the previous outbreaks? I know of some in California, but off the top of my head, I'm not going to be able to give you a complete list, but they were sporadic and relatively widespread. I'm sorry that I can't give you a more specific answer.
Sorry, another question. I've worked up and down the coast and spent time Haida Gwaii and Central Coast and Northern Vancouver Island, and there's like a shocking difference in terms of like Northern Vancouver Island. We saw like one sunflower sea star this summer and like one ochre star, and yet last year we got to spend time between Klemtu and Bella Bella, and it's the only place on the coast where I've seen abundant ochre stars. And is there something oceanographically in terms of currents and things like that that makes that area unique? Because even in Haida Gwaii we saw bat stars and leather stars, but ochre stars also were seeming to be quite rare. So.
Yeah, so that question is fabulous. It's exactly the thing I'm about to study. I guess starting answer, unfortunately, is I don't know quite yet, but I do work in that exact spot. I work on Calvert Island, between Bella Bella and Vancouver Island. And you're right, there are a lot of ochre stars there. One thing I will say is we're still missing the big ones. So we lost big ones, but the populations are quite large. So that, the picture of the pile of sea stars on a wall, it's actually from Calvert Island in the last couple years. So you're exactly right. They're all over the place. And maybe it has to do with that remarkable recruitment that we see there. It's potentially higher. So we found high recruitment everywhere. It's potentially higher there. I can't say that yet. But that is the exact question I want to know. what is different about that area and why. Are they doing better? Yeah.
For the younger starfish that aren't as affected by the disease, are they becoming more affected as they get older, or are they remaining resistant?
That's another great question. So I didn't talk about another aspect of the ochre star's biology that we do know about. There is a mutation in ochre stars that can make them resistant to disease. So this is a broader answer to your question than you asked. And so potentially some proportion of the population is surviving because of that mutation. And potentially if their offspring have it as well, they might be more resistant. So potentially they could grow up and be more resistant in the long run. It's a lot of potentiallys in one sentence. But we do also see some, I can say that I saw juveniles that were wasting this last summer. And I don't know why that was happening or how common that is either. So that's on the list of things that I'm really curious about. So in the past, it really seemed like the juveniles, they definitely are less susceptible, but I saw some pretty small wasting stars. So I'm not sure. Yeah.
Just wondering about the food source, and is it concentrated more in the food source, or do you find the different positive microbes versus negative microbes in the food sources themselves might be affecting the concentrations inside the sea stars, and therefore,
Yeah.
and I'm just wondering about how it's transmitted.
Yeah, so the question is about the food source and whether the microbes could be coming from the food source and if the food source could be sort of a driver of changes. Okay, what I remember is that there was a point in time where we thought that that could be how it was transmitted. Right now, our understanding of transmission is mostly that it's sort of point-to-point contact, so being near each other, and potentially through sediments. But I would say that we also don't know because there's sort of array of evidence that still needs to be thought through given sort of the new research I just talked about and different modes of transmission. So I'm not sure I can answer that question yet, but it's a really good point. Like, it's a place that we should continue to look.
So point to point is the main transmission.
Our understanding, yeah, direct contact.
Even if
And potentially, so this is what's hard to, why it's difficult to answer the question is that if for the virus, we've found the virus that's associated with some of the disease in sediments, in the water column, in filters when it's attached to an aquarium. And generally when they looked at sort of how long the virus stayed alive in the water column, it wasn't particularly long. And so the thought was that maybe it could transmit in the water, but not very far. But that's just for the one virus, and it doesn't really explain sort of wasting disease symptoms when we don't have the virus, and sort of that dysbiosis. I'm not sure we have an idea. We don't know yet how that's being transmitted, and if it's being transmitted, and if there's another virus that's associated or something, unfortunately. Yeah.
scuba diver, don't touch that.
Yeah, actually, yes. No, that's a fabulous question. So if you are out in the field and you are either a diver or just on the beach, I would not recommend touching one sea star and then touching another sea star because we could transmit it. We don't know. So you'll be fine. You can touch them and your person will be fine. But don't grab one and then go pick up another one. Just to be safe, we don't need to increase the transmission mode if that is one of them. Yeah.
Okay, I think we have time for just a couple more questions. Does anyone else have any more questions? Wait a minute.
Hi, have you seen the sunflower star populations increase or recover at all?
That's a great question. Up at Calvert, there is some evidence that the sort of small stars that survived wasting are getting bigger. So we still see the small ones and they're getting bigger. I can also tell you that, and this is very anecdotal, but I was out in the field this summer not proudly actually, sadly proclaiming that we were not seeing any recruiting juveniles. And that's generally what I thought. And then I turned a rock over and found one. So maybe they're there and we're just in the wrong spot. So I have found a couple of smaller sunflower stars, but in our actual transects, we're not finding them there. So yeah, but there are still sunflower stars, particularly further north in that sort of Calvert Island area. Elsewhere, they're very hard to find. Yeah.
Wait a minute.
Careful.
How did you decide what transects to go with?
Oh, so like how to set up where we study?
Yeah, how did you decide on the length and like the dimensions and that kind of thing?
That's a fabulous question. So generally where we selected the location was in a place where there were a lot of stars. So when it was first started, these were intentionally picked areas where we find sea stars. And that's because we're going to go back there over and over again, so we want to be counting where the sea stars actually are. And since we're going to one spot over time, yeah, so it's potentially one of the higher numbers in a given rock area, if that makes sense. The length that we picked was actually, so there's two ways to answer this question. One is that there's a really big, broad coastal program looking at stars that you saw lots of data from, right? And they've been working for years, and so generally it's good to pick the same length that they use so that it's relatable depending on where you are. But the other way to answer the question is also to make sure that you get that there are enough sea stars in that area so that we can see changes in their populations. So you don't want to go somewhere where there's only 5 and only count this very small area where there's 5, because it'll be hard to tell what happens to that